Greetings everyone. Today we will perform a differential expression analysis for bulk RNA-seq data using PyDESeq2. We will go step by step through the workflow, from loading and pre-processing the count and metadata files to modeling gene expression, performing statistical tests, and interpreting results. You don't need to look for any external dataset because PyDESeq2 provides built-in example data, so you can easily follow along. Without further ado, let's get started. We start by importing our libraries. To perform a differential expression analysis, PyDESeq2 requires two types of inputs: a count matrix of shape number of samples versus number of genes containing read counts, and the metadata or column data of shape number of samples versus number of variables containing sample annotations that will be used to split the data in cohorts. So to illustrate the required data format, we load a synthetic example dataset that may be obtained through PyDESeq2's API using utils load example data. So and we create also the metadata. Now let's print our counts data frame. Let's run our code. And here we have it. As you can see, we have columns of genes and rows of samples. In PyDESeq2, the input format is correctly structured when each row represents a sample and each column represents a gene. This organization is standard for Python's pandas data frames, so you don't need to transpose your count data before initializing the DSeq dataset object. Let's print our metadata. And here we have it too. As you can see, we have samples and we have two columns, condition and group. Condition and group representing two types of bi-level annotations. In the first part, we will only use the condition factor, and later we will see how to use both condition and group. We will see that in the multifactorial analysis. Next, we have to make data filtering. Data filtering is good practice to pre-process our data. For example, remove samples for which annotations are missing and exclude genes with very low levels of expression. This is not necessary in our case because we are using a synthetic data, but don't forget this step if you are using real data. So we will start by removing samples for which condition is none. So let's run our code. In this case, where the design factor contains NAND entries, PyDESeq2 will throw an error when initializing a DSeq dataset. Next, we will filter our genes that have less than 10 read counts in total. Note again that there are no such genes in this synthetic dataset. Let's run our code. And now that we have loaded and filtered our data, we may proceed with the differential analysis. So firstly, we perform single factor analysis. In this first analysis, we will ignore the group variable and use the condition column as our design factor. As you remember here, in our metadata, we have two columns, one for condition, which has variables a and b, and one for group, x and y. So that is, we compare gene expressions of samples that have condition b to those that have condition a. We start by creating a DSeq dataset object from the count and metadata. So as you can see, our DSeq dataset has counts for our row count matrix, metadata for sample into with the condition column, and design equal condition, which model formula use condition as the factor for differential expression analysis, and refit cox equal true, automatically refit samples flagged as outliers based on Cook's distance. And lastly, inference, this to use the multi-core inference setup. Let's run our code. And now, once a DSeq dataset was initialized, we may run the DSeq2 method to fit dispersions and logfold changes. So, let's run our code. And here we have it. Now, let's print our DDS. And we have it. When we print the DDS, we see it has 100 samples in rows and 10 genes in columns. In annotation data are stored in key-based fields, ops or observations, sample level data like condition, group, and size factors, var, which are the variables, gene level data like dispersions, and ops or observation matrices, multidimensional sample data such as the design matrix, var or variable matrices, multidimensional gene data like logfold changes. So and data or annotations data keeps all information, counts, metadata, and model results well organized in one object. Hence, parameters are stored according to the annotation data's data structure, with key-based data fields in particular. So, for example, if we want to access the dispersions, we write print DDS dot var, and we put dispersions. And here we have it. As we know, that dispersion measures how much counts vary between replicates for a given gene. In RNA-seq, it reflects biological plus technical variability beyond what's expected from the Poisson noise. Each value corresponds to one gene natural scale, not log. So, for example, in gene 4, it gives us 0.15, which means low variability and stable expression. But for example, gene 6 gives us 0.97, which means high variability, more spread across samples. So, higher dispersion means less statistical confidence in log-fold change estimates, while lower dispersion means the gene is more consistently expressed. And now let's print the log-fold change. So dds dot var m, and then we put log-fold change. Let's run our code, and here we have it. So as you can see, we have genes, intercept, and condition. For the intercept, it is the baseline expression level for condition A, the reference group. And for condition, it is the log2 fold change for condition B relative to A. It shows how much expression shifts when moving from A to B. So for example, gene 1, it's about 0.44, which means this gene is slightly upregulated in B compared to A, while gene 3, negative 0.43, it means that this gene is downregulated in B, and so on. So now that dispersions and log2 fold changes were fitted, we may proceed with the statistical tests to compute p-values and adjusted p-values for differential expression. So we write ds equal to So as you can see, DDS, which should be a fitted PyDESeq2 dataset object, and contrast, which is a list of 3 strings of the form variable, tested level, and control level, or directly a contrast vector. It also has a set of optional keyword arguments like alpha, Cox filter, and others. So let's run our code. And now we can perform the wall test, as PyDESeq2 computes p-values using wall tests. This can be done by using summary method, which runs the whole statistical analysis, Cox filtering, and multiple testing adjustments included. So we write ds.summary. Let's run our code. And as we can see, running wall tests now. And here we have it. As you can see, here are the genes, base mean, log2 fold change, log fold change, standard error, stat, p-value, and adjusted p-value. We can also go for threshold-based tests, as we can specify a log fold change under the null hypothesis and an alternative hypothesis to recompute wall tests and p-values. The alternative hypothesis corresponds to what we want to find rather than the null hypothesis. So and here we have it. This runs the whole test, but now tests if genes have an absolute log-to-fold change greater than 0.1. And for the log-fold change null equal 0.1, here we set the threshold under the null hypothesis. No meaningful change if log-fold change is smaller than or equal to 0.1. And here alt hypothesis greater abs. It looks for genes with log-fold change larger than positive or minus 0.1. So in short, it finds genes with biologically meaningful expression changes, not just tiny ones. And then our MA plot. As you can see, x-axis is for the base mean or the average expression, and the y-axis log-to-fold change. Each point is a gene. So for example, this gene is upregulated in B, while this gene is downregulated in B condition, and so on. For visualization or post-processing purposes, it might be suitable to perform log-fold change shrinkage. This is implemented by log-fold change shrink method, which takes as argument the name of the coefficient to shrink. For example, the name of one of the columns of the design matrix. For instance, to shrink the log-fold change of the condition B samples, we would run this code. So ds dot log-fold change shrink, and we put coef. Let's run our code. And here we have it. This is shrunk log-fold change and wall tests. We have the base mean, log-to-fold change, log-fold change standard error, stat, p-value, and p-adjusted value. Note that running log-fold change shrink will override a DSeq stats log-fold changes with shrunk values. This can be checked using shrunk log-fold change flag. So if we write, for example, print ds.shrunk log-fold change, let's run our code. We got true. And now let's go for multifactorial analysis. So far we have only used the condition column of the metadata, which divides samples between condition A and condition B. Yet metadata contains second column which separates samples according to group X and Y. For example, metadata. As you see, here we have group X and Y and condition A and B. We have used condition A and B before, but now we would like to use both columns, condition and group. So the goal of multifactorial analysis is to use both variables to fit log-fold changes. So to perform multifactorial analysis with PyDESeq2, we start by initializing a DSeq dataset as previously, but we provide several variables we would like to use in the design argument. So we put group plus condition. As you remember, before it was just condition. And now and here we have it. Now if we print log-fold change, we will have two columns in addition to the intercept, one corresponding to the group variable and one to the condition variable. So let's try. Let's run our code. And here we have it. As you can see, beside intercept, we have group and we have condition. One for the Y and one for the P. So, as you remember, here the design was group plus condition. So, this tests condition A versus B while controlling for differences due to group X and Y. So the model explains variation in expression using both variables. So for example, here, this shows to us the intercept here, which is the baseline group X and condition A. And here, group T and Y, this is the effect of being in group Y. While here, the effect of being in condition B. P-values are computed as ELIA from a DSeq stats object with the summary method. The contrast argument will allow us to determine which variables we want to obtain log-fold changes and p-values for. So, the first one is the variable, and then the tested level, and then the reference. So, for example, ds b versus a equal to DSeq we put the first one, the variable, tested level, and reference. And then let's run our code. Now let's fit p-values. And here we have it. As we can see, although we are comparing the same cohorts, which are condition B versus A, the results differ from single factor analysis. For example, in gene 1, the p-adjusted value is 0.02 for gene 1, while here is 0.064. And this is because the model uses information from both the condition and group variables. Let us now evaluate the differential expression according to group Y versus X. To do so, we create a new DSeq stats from the same DSeq dataset with contrast, group, Y, and X. And then we run our analysis again. So, let's run our code. And here we have it. So as you can see, here for the condition and here for the group. And as we see, the numbers are different. So for example, here 0.026, but here is 0.020, and here 0.00, and here 0.44, and so on. In a multi-factor setting, log-fold shrinkage works as in the single-factor case, but will only shrink the log-fold changes of a DSeq stats object based on its contrast argument. So, and here we have it.
This step applies log-fold change shrinkage to the condition B versus A contrast using a Bayesian method. It pulls the extreme log-2-fold change values toward the more moderate ones, reducing noise and producing more accurate interpretable differential expression results.
And here we've reached the end of our analysis. If you'd like us to make a tutorial or episode about another topic, feel free to leave your suggestion in the comments below. Don't forget to like this video. Thanks for watching. See you next time.